Today we have learned the title of the mechanism of separation. How the separation takes place. How the proteins are migrated in the stacking gel. How the proteins are migrated in the separating gel. What are the phenomena where takes place. So those are information today we have learned. Okay, first we started with the mechanism of Sds Page. You know very well, discontinuous buffer system is more suitable for separation of protein, especially in the case of SDS. I'm already told two type of buffer systems are used in the phase polyacrylamide electrophoresis, either continuous or discontinuous. But we are more focused to discontinuous buffer system. Why? Because the proteins are highly soluble. Okay. Moreover, pH dependent gel. The adverse electrophoresis consists of single pH. We are using 8.2. But here we are making different type of pH gel. Either stacking gel is 6.8, the separating gel is 8.8. The basic difference between the gel is the pH. Actually, the pH 2 is differ. 6.8, 8.8. 2 pH is difference between the gel. Number 2, the concentration of polyacrylamide solution. Either stacking gel is nearly 5% and the separating gel is 10%. So these are the main difference between the stacking gel and separating gel. Moreover, regarding the concentration of polyacrylamide, so the pore size is increased in the stacking gel compared to separating gel. Separating gel having smaller pore size. So you recall this information before studying of the entire mechanism of Sds Page. Okay. So listen carefully. Now I am conveyed the very important mechanism behind the Sds Page. Okay. Okay. Before starting this mechanism, you try to know the name of the word is counter ion, leading ion, and tailing ions. So this is the important terminology we are using here for studying. What is called a counter ion means, so those ions are encountered during separation. The counter ions are divided into two types. One is leading ion and tailing ions. Two type of ions are there. Okay. So the leading ion means the ions, any type of ions or a particular ions move faster than the fast moving protein, then the slow moving protein, and then the tailing ion. So these ions are migrated very fast. It's called leading ion. Tailing ion means the ions are migrated but very slow, less than the leading ion, fast moving protein, and medium moving protein, or slow moving protein. So these ions are last ion get migrated very slow. This is called tailing ions. Okay. So these are the terminology we are going to discuss. You just imagination what I am trying to tell. Just imagine the gel. So just you are going to sit in the inside of the gel. Yes, inside the gel. Go to sit inside the pore. Then only you completely understand what I am told. Okay. The discontinuous system is suitable for Sds Page. Okay. It contains two type of gel. One is stacking gel. The discontinuous employs two buffers. Yes, two buffers with the pH differences 2. Because the stacking gel pH is 6.8, separating gel pH is 8.8. And also the large pore size with the stacking gel is there. Similarly, the separating gel pore size is very small. Okay. When the electrophoresis started, just imagine when the electrophoresis started, when the voltage is applied. The time, first, when the voltage reached the electrode around buffer containing water molecules get hydrolyzed. You know very well. Hydrolyzed, finally produced H plus ions and OH minus ions. Okay. So those and the H plus ions try to move the corresponding electrodes. Okay. This is the first reaction takes place. Followed by this hydrolysis, we are supplying the tris HCl as a part of the buffer, a part of the running buffer is called a tris HCl molecule. This tris HCl get dissolved in the water. It produced tris and hydrogen and chloride ions. This chloride ions is called leading ion. You know very well, chloride is highly negative, so it can migrate very fast to the anode. Followed by we are supplying the glycine molecule in the sample loading buffer. Okay. That sample loading buffer containing glycine molecule get ionized in the stacking gel at the pH of 6.8. You know very well, glycine is the basic amino acids get ionized and form a zwitter ion. If the glycine get ionized, what's happening? The excess, because 6.8 is the high acidic pH, so it's contain huge amount of H plus ions. So those H plus ions are added to the amino group of the glycine molecule. So this glycine molecule converted into glycinated ions. Glycinated means huge amount of positive charge there in the glycine molecule, especially in the carboxylic group, especially in the amino group. So it's exhibit NH3 plus here COOH minus. So it's highly basic in charge. But the stacking gel to forwarded to especially the cathode. Okay. Yeah. So the glycinated ions are move very slow because it's highly positive nature. The positive nature of this glycinated ions highly attracted to the cathode because cathode is negative, ions are cations, are positive ions. Okay. But it's move very slow because cathodes are attracted this glycinated ions when it's present in the stacking gel. Okay. Similarly, chlorine molecules are highly anionic form, which fast move to anode. Okay. So the glycine ions are tailing ions, chlorine ions are leading ions. Okay. Yesterday I told, so finally glycine as a tailing ions, chlorine molecule as the leading ions. Okay. Okay, in the stacking gel portion, you just imagine the stacking gel alone. Okay. So stacking gel is the high pore size, pH 6.8, and also we are placing the sample. Yes. And also the stacking gel contains SDS also. So those SDS molecules are highly denatured the protein, sample protein molecule. Okay. Just imagine in the stacking gel portion alone, I only try to tell the stacking gel alone. So the bottom of the stacking gel is seriously arrange the chlorine ions first. When the voltage apply, the bottom of the stacking gel was highly arranged the chlorine ions. Followed by chlorine ions, the fast moving proteins are arranged, second phase. And the third, followed by the fast moving protein, the third part of the layer is medium migrating protein. Then followed by next, slow moving proteins are stacking. Then followed by glycinated ion. This is very slow moving ion. This structure look like a sandwich. You know sandwich? The first sandwich is a bread, so chlorine ion. Second one is cheese. Cheese is the fast moving protein. Third, cabbage. Cabbage is the medium migrated proteins at stacking. Followed by cheese, the slow moving protein. Followed by bread. Bread is the glycinated ion. You look like a sandwich. Yes. This process takes place happening at the stacking gel area. Even applied the voltage, the proteins are look like this. After some time, maybe around 30 minutes of time. Okay. Because the pore size is high, so no distractions are there. So this seriously arranged. Okay, this setup then immediately enter or encounter to the separating gel because we are applying voltage. Okay. The time this chlorine ions are completely ionized, irrespective of pH, is the fast moving become a leading ion. Is the mobility is faster than the fast moving protein as the ionization of glycinated ion? Yes, because glycines are ionized at the pH dependent. Yes, because now the glycine molecule placed in the 6.8. Yes, so it have some positive charge ions. So it's called poorly ionized at the 6.8 pH in stacking gel at 6.8. It becomes a slow moving ion. This setup placed in the stacking gel, this setup, its mobility is slower than the slowest moving protein. Okay, yes, compared to the glycine, the glycine's mobility is slower, very slower than the slowest moving protein, because the glycines are standing at the last row, last horse, last individual, last things at the line, okay, at the sandwich. This setup produce a very high localized voltage gradient. Yes, this highly localized voltage gradients are setting between the fast moving chloride ions and the slow moving glyceride ions. Yes, so between the fast moving chloride ions and slow moving glyceride ions, between these the fast moving protein, medium migrated protein and slow moving proteins are arranged between them. Please keep in mind. This process will create some localized voltage gradient. Very important term is, since the protein have intermediate mobilities get stacked between the leading tailing ions. Yes, this setup is called isotachophoresis. Isotachophoresis is the arrange ready to migrate the ions and proteins in the stacking. So the stacking gels contains, it's not an electrophoresis. This is called isotachophoresis. Okay. Then the setup immediately enter into the separating gel. When they reach the separating gel at the pH of 8.8, the glycinated ions are completely ionized. Yes. Why the glycinated ions are completely ionized? Because it's entered into the pH 8.8 in the case of acidic. So the chlorides are move faster in the separating gel also. So if the glyceride ions are enter, that means the entire setup transfer into the separating gel. So the glyceride ions are get ionized. So it migrated more faster than the slow migrated protein, medium migrated protein and fast migrated protein. So it goes to sit below the chlorine ions, Then you just compare to the separating gel. So here setup is chlorine, glyceride ions, fast moving, medium migrating and slow migrating proteins are arranged like this. Yes, because glycines are highly ionized when it's enter into the pH 8.8 at the separating gel. Okay. These two ions, especially chlorine ions or glyceride ions, start move faster than the proteins at the separating gel. The time the chlorine ions or glyceride ions followed by glyceride ions to create a path for migration of fast migrating or medium migrating or slow migrating proteins. It's create a path to allow the migration from cathode to anode. So that time that fast moving protein or medium migrated protein or slow moving proteins are separated according to their shape, size and molecular weight. So this is happening. This process was happening at the stacking gel. Okay, you know very well proteins are colorless. So how we are identified where it's migrated, whether it's migrated or not, where it's migrated, and also we are calculating RF value also. So for this purpose we are adding Kamashi blue dye into the sample in the form of sample loading buffer. So we are adding sample loading buffer sample loading buffer containing commercial blue. This sample loading buffer added to the sample. Okay. So this commercial blue will indicate where the proteins are migrated, either is migrated or not, either across the interface between the stacking gel and separating gel. Once this commercial blue to reach the anode, we can indicate, we can decide either proteins are migrated. This is the easily visible things. So this is the image we are placing here. This is stacking gel, 6.8 glycinated ions, 12. The pH 6.7 because glycine pH is PA is 6.07. Yes, it's very slow ionized at the time of when it's placed in the pH 8 or stacking. Suppose when across in the separating gel, it can migrate more faster because the ionization is highly ionized. This is the basic pH. Okay. Followed by proteins are separated. This is the phenomena happening at the gel. Yes. So please keep your mind.